Good morning, good afternoon, and good evening to the members of the Pasteur Network community, and welcome to this new edition of the Pasteur Network monthly seminar. Thank you very much for joining us today. We will begin with our speaker presentation, followed by the Q&A session. Feel free to post your questions and the chat at any time or during the Q&A. To ensure a smooth experience for everyone, please keep your microphone and camera off during the talk. The seminar is recorded and will be soon available on our Pasteur Network YouTube channel. So let's begin. With the rapid rise of AI over the past few years, it can be difficult to know how to position ourselves as researchers, health professionals, and global health networks like the Pasteur Network. This is why we've dedicated this monthly seminar to this topic. Today's session focuses on AI-assisted microscopy for clinical decision support as a cornerstone for global health collaboration. And for that, it's our great honor to introduce our speaker, Joe Schott, Chief of Global Strategy and Partnerships at Enaiblers. Joe is a career global health practitioner and R&D portfolio manager with over 20 years of international experience spanning USAID Global Health Bureau, the National Institute of Allergy and Infectious Diseases, the Walter Reed Army Institute of Research. He has also served in numerous WHO advisory roles and co-designed key initiatives, including the WHO NTD Diagnostics Technical Advisory Group, the NTD R&D Blueprint, and the Expedit Review Panel for NTD Diagnostics. Following the close of USAID, Joe joined Enaiblers, a Swedish global impact meditech startup, and chief of global strategy and partnerships, bringing with him deep experience working alongside local research institutions, universities, ministries of health, biopharma industry, and multilateral organizations. So before handing over to our speaker, just a very quick word on PNLink, our knowledge sharing platform. So the PNLink platform has been launched 7 months ago. PNLink is already having more than 1,200 active users from the Pasteur Network. Almost 95% of these users are researchers and public health experts. And you are all using it, and we are really grateful for that. So just want you to know that if you're not on PNLink, think that PNLink is completely free and open to all the members of the network, including researchers, students, and support staff. We are creating more and more groups, and we are planning to create a group on AI applied to health very soon. So yeah. Let's keep in touch and keep following us on PNLink, and you will know more and more about knowledge sharing and AI tools for the Pasteur Network. So I will let now Joe share your screen so it's already done. Thank you so much. So the floor is yours, and thank you for being here.
Great. I just wanted to confirm you can still see my presentation mode.
Yeah, I'm sorry, I was muted. Yeah.
That's okay.
See it.
Okay. Very good. Thanks, Hakim, for the really warm introduction. I'm really pleased to be here to represent Enaiblers and to meet everyone. Globally, it's quite a diverse network, and it's really exciting for me to be able to be here, and really thank you for that. Just to give you a background on who we are. So we were previously working within Johnson & Johnson, so the global pharmaceutical company, and one of the main programs that donates deworming medications for preventive chemotherapy. And one of the social impact ambitions of the company was to really improve the diagnostic capacity and thus the data for monitoring and evaluation for these large-scale disease programs. And so in 2022, Enaiblers spun out of Johnson & Johnson and established and founded a headquarters in Uppsala, Sweden. And so the overall mission for us is to make AI-powered imaging accessible to anyone, anywhere. And in thinking about all of the challenges and why we focused on this one was really clear initially from J&J and other companies that improving data needs for countries to continue to access these drug donation programs was increasingly being a barrier. And also to think about in a post-drug donation world, what would the key competencies be to ensure that countries could still understand the epidemiology and the impact of some of these diseases of poverty. And then as we've seen more recently, there's so much less global health assistance funding from donors, and that's really kind of accelerated the need to integrate programs. And when I say integrate programs, we even think about potentially maternal child health, nutrition, and infectious diseases as being integrated, not merely integrating two different infectious diseases. And really thinking about those workforce efficiencies that will have to change as a result. If you think of really large, high-burden countries, the standardization needs across that country are incredibly important. It's been our experience, from my experience working with USAID, that new test procurement, whether it's molecular testing, antigen detection testing, antibody detection, those new tests are really difficult to procure, especially for NTDs. And I'll get into that further along in the presentation. And then just lastly, one of the main challenges is in line with the NTD 2030 roadmap of WHO, the M&E frameworks overwhelmingly retained some component of parasite and vector analysis for confirmatory testing. And thinking about the focality of disease transmission and dynamics, we know that parasites and vectors don't respect administrative borders or geographic borders. And so really equipping the workforce, whether that's a laboratory technician that doubles as a community health worker or a clinical trialist who's a principal investigator, equipping them with something that is of high quality and can provide that assurance, especially at these really low transmission settings. And as one example here in Sierra Leone, with applying a geostatistical surface across the country, which ignores administrative barriers, the health system really has a whole host of different intensities of infection, and this is capturing really just one disease condition, and that's soil-transmitted helminths. So in most everyone here who has been working on the ground in endemic settings for whatever disease profile that your institutions are trying to address, microscopy is still one of the foundational cornerstones of screening and diagnosis. It's quite adaptable, it's well established, and it allows for direct visualization. However, there's increasing limitations to this, some of which I've previously covered, but the primary ones that are widely recognized as the inconsistent quality, the impact of sample preparation on the quality of the reading, frequent need to retrain expert microscopists, and I think in the malaria and the TB community, this is something that's been really addressed head on to really bring that quality up. And so a solution that we have developed and have started implementing with partners is this concept of AI-assisted microscopy. And so really, essentially what this does is there are no major changes to the sample preparation step, which is what the workforce has been trained to do. And so we have an AI-assisted platform that uses machine learning to, and in this case, when the microscopy slide is entered into the device, it does the automatic boundary detection, and in this case of a smear, the scanner then does the automated scanning, and then the model does the image analysis and provides results in the form of some sort of either desktop or mobile app for the output, and then this can feed either into epidemiological research or health data. And so really what we're talking about is this type of setup here. So this is from our collaborators with the Ministry of Health in Uganda. Here we have multiple platforms set up in the context of a national soil-transmitted helminth prevalence survey. As you see, we are hooked up to car batteries. The platform can be able to have battery packs, to have solar panel connectivity, and it also has on-platform storage for images, and then, of course, Wi-Fi upload when that is available. And the important thing here is that there's still the need for human in the loop interface. And so this is not intended to diminish or replace the workforce, because the quality management system is still incumbent on individuals to use this tool as clinical decision support and verify the findings of the AI. So really, one of the primary main pain points has been addressed through development and deployment of this platform, and that's the automated and integrated approach. To date, we have either done pilot evaluations or have clinical validation data in a host of different diseases and biospecimen types and preparations. Cato cats for some of the parasitic diseases, blood smears currently for identifying and quantifying Wuchereria bancroftian filariasis, direct smears, which could be for a whole host of different intestinal parasites, urine filtration, and some exploratory work with tissues and entomology. But as with AI and machine learning, is the beauty of it is that it's quite agnostic, and it can really be used for a variety of different kind of parallel fields. And so whether that's animal health, veterinary health, environmental monitoring, material science, and life sciences. And so really the possibilities to scale the concept up for a variety of different conditions and both imaging and specimen analysis is pretty wide open. We've been working with different sorts of collaborations globally. So, key are our research collaborations in endemic settings, and they're really those on the ground who are stakeholders that provide input to the challenges that they're facing and really what will aid them in making their jobs and their lives easier with the tasks at hand. And so we have been underway in about a half dozen countries and recently had some publications on those findings. We, of course, have been engaging with national NTD elimination programs, such as those for schistosomiasis and soil-transmitted helminths. And then, of course, some of the performance evaluations on the model itself. I wanted to jump into a series of examples from countries and case studies to give you an idea of our approach and some of the implementation research that we've been undertaking. So in these settings in Peru, Ethiopia, and Uganda, we've conducted specimen collection, scanning, and analysis in a variety of different levels of infrastructure settings. More recently, we finished some work on Cato cats specimens for soil-transmitted helminths in Uganda and Ethiopia. And the goal here was to supplant the platform into a routine established national prevalence survey. So this really mirrors the routine practices of the community health workforce that's engaged in these activities. And what we found is that, due to the virtue of the platforms being more sensitive and the consistency does not vary from read to read, and the avoidance of fatigue in reading something like microscopy slides all day or all night, the verification data was quite tight and quite impressive. And here we've got a graph from roundworm infection, looking at eggs in stool. And so just a closer up, the agreement, as expected, is quite tight as you get higher in numbers, and then does separate a tiny bit at the bottom. But the key here is that machine learning, AI does not fatigue, and the consistency is really maintained. And so here, if we look at across the species, both at low intensity infections and medium and high intensity infections, the AI outperforms human readers in all instances. And this is really important in some of those low prevalence settings that I mentioned, where maybe the quantities are extremely low and may go undetected. An initial lab evaluation that we did with our colleagues at the University of Queensland in partnership with one of the Pacific Island nations was to take a characterized data set of blood smear results in a Wuchereria bancrofti elimination setting. And in those 100 slides, the agreement was quite good. The key thing here, though, is in the lower left image, there was a single microfilaria on this slide, and 2 expert technicians did not detect that and assigned a diagnosis of negative. And so it was actually the AI that picked up the one microfilaria in the sample, and thus this individual was walking around transmissible. We've recently submitted some data comparing 6 different established methods for schistosomiasis, the hematobium species. And so looking across the different analytical methods, the AI is really right up there with qPCR. I think that, since the qPCR target is obviously molecular and may not always be found in sample preparations where the eggs are intact, we think that that's potentially the reason for such a high performance compared to PCR with the AI. Shifting to another disease, in collaboration with our colleagues at CDC, we did an analysis of images. So these are not tissue sections that we had direct access to. This is training AI to look at established image banks. And so here, the concept is that we can train the AI to reproducibly and accurately identify mycetoma in images from tissue sections. So overall, the pipeline that we have ongoing is that we have quite good data and are quite confident in the areas with the green tick mark. We have a lot of work in process to think about how we may increase magnification. One thing to notice that we're currently only at between 10 and 20x magnification for the platform. So that does currently exclude the smaller pathogens, such as Plasmodia, TB. And so for now, we are, comfortably in the detection range of 10 to 20 and have some quite good validation data for those. And so in thinking about this presentation and thinking about, how we can collaborate and think of pilot projects together, we were approached by the Carter Center, which is one of the leading organizations globally in disease elimination, particularly for guinea worm disease, for river blindness, onchocerciasis. And so the problem is that was presented to us was that there are so many needs for Simulian black flies to be collected, sorted, classified, and then processed for PCR. And so in the context of the river blindness program globally, the 3 phases that really necessitate this are mapping communities to identify and establish those first-line villages that are going to receive ivermectin mass drug administration. Then the surveys to stop those treatments, to monitor the progress under those drug treatments for several years, and then 3 to 5 years of requisite post-treatment surveillance. And so if you think of the scale, which this is now principally solely in sub-Saharan Africa and in Yemen, and in a tiny focus on the Brazil-Venezuela border, the workforce is dwindling for these experts to be available to do these surveys and process the specimens, and there's just hundreds of thousands of flies annually in sub-Saharan Africa that require this. And so the engineers in Enaiblers, working with partners in Ethiopia and with the Carter Center, did some modifications to the engineering to develop a special holder for the vectors to then process through the AI model development and training on the data. So what we were able to do is create these 3D printed black fly holders in-house. The black flies are then loaded into the holders. And then those sample preparations can then occur in the laboratory by loading the cartridge, or the holder rather. Those are then inserted into one of the slide scanners. And here we have an example in Ethiopia of a lab technician operating 4 devices. So you really then get the scale of what's possible. Then the machine does the automatic smear boundary detection, and then the automated scanning, and the and the automated image analysis. And then we get results that can be presented on a laptop or on a smartphone. In this instance, it's on a laptop. And then this allows the technician, as I mentioned, it's important to have human in the loop verification of the AI data. And so having it in an interface where those that are reviewing can refuse the image for issues of quality, or provide the quality check on the speciation of the vector. And then here we have an example of the different indicators and fields for the image and how the individual can access those. So as an example, I have one of Similium bovis, which is one of the black fly species that needs to be characterized before the qPCR analysis. And what the imaging allows is for AI to use characterized features on the legs of the vector to actually assign a species to that fly. When we think about operational efficiency, as I was mentioning, for the black fly analysis, we're around the same efficiency as a human, but when you get to operating multiple devices, you actually see the scale and the cost efficiency as you increase the number of scanners. And one technician can work the full shift on doing multiple devices, and so we actually get up to around 80 flies that are analyzed per hour. And that's everything from loading the device, the speciation, and the data output is all able to be conducted in that time frame. Another aspect that we've been working on is thinking about different parts of the health system and looking at other neglected tropical diseases initially. And so we recently received funding from the Global Institute for Disease Elimination in the UAE, working with our partners in Madagascar and the Carter Center on a semi-integrated multiplex for diagnosis of SCH, schisto, LF, and tapeworm in high-risk families that raise pigs. So here we have a brief outline of our semi-integrated in such that we're piggybacking on some other studies and also embedding ourselves within the National Elimination Program activities. And so really here the goal is to work out those workforce challenges, sample preparation, and sample custodianship challenges that we may face in a variety of different settings. And so really to inform really good operating procedures and SOPs going forward for a variety of diseases. We recently were notified that we received 2 years of grant funding to continue to develop the version 2 of our platform in 6 countries with ISO, this is a typo 13485 compliant quality management system so that we can do small-scale manufacturing in Sweden and work with our partners in Peru, Brazil, Cambodia, Laos, Zanzibar, and Uganda to really develop those platforms for schisto and STH in line with pursuing IVDR and also the more NTD-specific regulatory pathways at WHO, which I'm going to briefly go into here. So it comes in no surprise that for many of the neglected tropical diseases, especially the ones that are not that don't have high mortality, is that diagnostics are not really a moneymaker for private industry. It was also apparent in the landscape analysis that that we at USAID at the time did with our colleagues at WHO is that there's really a whole host of different homebrewed and procured from a variety of different manufacturers around the globe diagnostics that don't necessarily meet the quality that's needed and also at the same time don't meet the benchmark for full pre-qualification review. And so one of the things that we established in 2019 in partnership with WHO was the NTD Diagnostic Technical Advisory Group. This included disease vertical groups to take up the challenges in the roadmap for diagnosis and screening of NTDs. And so there were expert groups by disease, as well as those in the horizontal cross-cutting areas that are equally important that underpin all of the success. So lab capacity, manufacturing and regulatory, imaging and surveillance platform. This group is meeting again in June to revisit their appropriateness for the ambitions that were set out in 2020. And my understanding is they're going to take up some of the new topics such as AI and machine learning in the space of diagnostics for NTDs. It's been really important for us. In addition, as I mentioned, we're working also through and with national elimination programs is that we meet these WHO TPPs for the different diseases. So making sure that we're not under or over estimating the work that's needed to achieve that. And so it's been really important through collaborative action and through listening to our partners and engaging with WHO that we really have aligned products and aligned technology for use by a variety of different end users of the platform. Briefly on the kind of entry into the market, this is a process that we worked with at WHO on called the Expedited Review Panel for Diagnostics. This is a modified process out of the PQ unit at WHO, whereby diagnostics that are deemed by consensus as being very low risk to an individual and or are not the sole decision factor for initiating an individual on medication or drug treatment, that these are handled as a risk classification A or B under this scheme, and thus they would not require the 2 to 3 years of WHO pre-qualification process and the higher cost of bringing those types of diagnostics, which already don't have a high market return on investment, to companies to really get those into the hands of not only the national elimination programs, but also researchers. I think just in ending, I wanted to give a quick snapshot of our very lean and mean team, which the original enablers, Lindsay, Levin, Peter, and Peter, are here. So we're a small team of engineers, of public health practitioners, and academics that make all of this possible. And of course, it's not possible without all of the collaborations that we have, with all of the input that we've received from numerous endemic countries, from individuals and research institutions, and the real strong support from WHO, both in the regional offices, but also at headquarters. And then just quickly, just to end, some of our enablers, so funders, technical partners, and research institutions that we've been collaborating with for the past few years. And with that, I thank you. And I also wanted to convey that should anyone have any questions on AI, I am not the AI expert. I will relay those to those that are experts either in engineering or in the AI models themselves. But I'm happy to take any questions about the work that we've done to date to the best of my ability. Thanks. Thanks so much, everyone.
Thank you so much, Joe. Okay, so thank you. So great presentation. I think that at many levels, it's very useful for the network of the Pasteur network as a network, but also for the members of the Pasteur network at the local level. So okay, so we open the Q&A session. I already have some few questions, but
yeah, great.
I would rather to have a question from the audience. So the floor is yours. Yeah, please, Kathleen.
Hi, Joe. Thank you for this very interesting presentation. I have two questions. The first one, you say that you give the machines the samples with flies or helminths, and then you check what AI does, which is what we all do and what we all have to do with AI at this stage. Do you have any idea or what's your opinion on, do you think that humans will always have to check after a certain time, or do you think that AI will be as performant as possible and that the human checking will be not needed after a while? That's my first question. And my second question is, lots of our institutes also work on malaria. Is that something that you plan to extend to? I saw that in the WHO group, there is something about African trypanosomiasis, but what about leishmania? What about malaria? Is that planned, or do you have collaborations on that? That's it. Thank you very much.
Thank you, Kathleen. So for the first question, well, you've hit one thing right on the nose. As AI gets better and as the data set gets larger, that sensitivity and specificity will always keep getting higher. It'll always keep getting better. But that said, without knowing the future, really, we still view the human in the loop as essential, especially when it involves interacting with any part of the health system where individuals or communities may have an intervention, whether that's a drug treatment, mass drug administration. So I still think that there's currently a need for human in the loop interaction in these diseases that we're working on right now. I think that in animal health, that would probably be a bit more wiggly. I think there's a bit more leeway with automation, and my understanding in the private sector in North America that this is already happening without humans in the loop. But I still feel that as this is, I wouldn't call it nascent, but I still think as this is moving from proof of principle into larger scale implementation for these NTDs, that we envision a human staying in the loop. And also, the platforms they're quite robust, but they require care. They require, gentle cleaning. They require careful transportation. And so we can't completely separate the human from the quality management system and from the technology itself. So it's a really hard question to answer, but we see the human in the loop to stay for the foreseeable future. Everyone asks about the malaria question. It's a good question. We're limited by bandwidth and funding to pursue higher magnifications. As with most things, as you get to a higher magnification and having multimodal lens magnification within device, the price goes up. So I think there's a lot of interest if we could find the right funding partner and collaborators to explore Plasmodia. We already are at the magnification to do the trypanosomes, and I've been really keen to look with partners, like DNDi and others that are trying to quantify the number and speciate different bloodborne parasites. So I think for as far as the sample type, we're not really limited. I think we'd have to look at the engineering and the whole ecosystem together, but certainly bloodborne parasites, the trips are completely within game. We could do that with the right partnerships. I think leishmania, my understanding is these are intracellular or also, okay, so that would, my understanding is that this would involve needing a high quality data set of images to train on the model, to identify them, supported by experts who can do the differentiation by species, whether it was Donovan or the others. And then, yes, that could be done. But we're just limited mostly by people's time and money.
Okay, thanks a lot.
Yeah, you're so welcome. Thank you.
Thank you, Kathleen, for a question. Any other question? Joe? So, okay, my turn now.
Yeah.
So, now you remember when you first discussed, Joe, about the specificity of our network, that the Pasteur network, so it's a network of research centers, public institutions, institutions working in the field of public health, working on the field, on the ground, in the labs, also producing vaccines, therapeutics, all these missions. How do you see a potential collaboration with a network like ours at both levels, at the global level, but also at the local level? I know it's quite complex as a question, but
yeah. Yeah, I have thought about this topic since we discussed it. I think that we really, because AI and because we are pretty agnostic about the type of use of the technology and the platform, we really want to hear about the challenges that institutions or countries or the network at large are facing. I think the low-hanging fruit is working as a QC resource, whether that is for training, whether that is for clinical trials or epidemiological studies. And so I think that really there's a whole host of different possibilities, for a collaboration. And we here in Sweden aren't necessarily wed to one style or one type of interaction. I think that the partners that have reached out to us have either had a workforce challenge, whether that's number of samples, the burden of sample analysis, or the data flow. We want to hear about those. And, we don't have a solution for everything and don't have an engineering solution for every type of analysis, but in just with Kathleen's comment, those are examples where we could certainly collaborate together and think through, what is the real problem we or with you are trying to address? Are there image analysis or AI or engineering tasks that we can do together to solve that challenge or to address that bottleneck? And those aren't something that we bring to the table knowing first. We want to hear from the ground up. And so I think that it's a good question, and not one I have a really specific answer for, because with some of the examples with the black fly analysis challenge, with the mycetoma example, those came to us from individuals and institutions, and they came to us with a challenge. And so we would love to be a sounding board. We would love to hear ideas about whether across the network, a plurality of the institutions are facing a challenge, or is it an individual country or country institution that's having a specific challenge where either image analysis on actual images or specimen analysis on discrete specimens can be wholly or partially solved by AI. And we'd love to hear for examples on that. I think another one that just came to mind, University of North Carolina in the US has a water institute. They are developing diagnostic for Naegleria filariae. For most all of you that know, this is the amoeba that can, in exceedingly rare cases, cause fulminant meningitis and death. We have a couple cases a year in the United States, and there's just no indication of how prevalent this infection is or its modes of pathogenesis. And so we're in early discussions with them to see, is there some sort of role, whether it's image-based or sampling-based or engineering-based, to help them with their sampling strategy for freshwater bodies in the United States. And so I think that technology is flexible. I think we're flexible, and so I think we need to hear the challenges and then work together to think if there's any potential solutions for those.
Great. Thank you so much. I think there are many opportunities for collaboration there. Yeah. And about Naegleria filariae, there is just for your information, the Institut Pasteur in Guadeloupe is working on that. Okay. So if you want, I can put you in contact with them.
You bet.
Yeah. Okay. So if we don't have any other questions, I want to thank Joe for your time and your presentation and this potential collaboration between Enaiblers and the Pasteur Network and the members of the Pasteur Network. And yeah, that's it.
Thank you. Merci à
tous. See you in a month. And once again, Joe, thank you so much.
Thank you, everyone.
See you. Have a nice day.
Thanks.
Thank you. Bye-bye.
Thank you.
Bye.